let's now move on to the next speaker. It is my honor to introduce our next speaker, Dr. Sachiko Isobe, Head of Laboratory of Plant Genomics and Genetics from Kazusa DNA Research Institute in Japan. Dr. Isobe started her career as a conventional red clover breeder back in 1997. She obtained her PhD from the United Graduate School of Agricultural Sciences, Kagoshima University in 2006, and has worked for Plant Molecular Genetics and Genomics in Kazusa DNA Research Institute since 2007. Currently, she is a laboratory head of plant genomics and genetics, performing de novo whole genome sequencing, molecular genetics, database construction, and digital phenotyping for plants. She is also a leader of Lab of Plant DNA Analysis, which provides contract-based services such as genome analysis of cultivated plants and related microorganisms and development of practical technology for their analysis. So today, Dr. Isobe will be sharing her expertise in the large-scale genotyping in strawberry genomic selection. I'll pass it now over to you, Dr. Isobe. The stage is yours.
Thanks, Joy. Great introduction. And hello, everybody. I'm Sachiko Isobe in Kazusa DNA Research Institute. And today I'd like to talk about strawberry genomic selection and how we use the large-scale genotyping systems in these selections. And this is a strawberry cultivation situation. And the strawberry production is actually increased very much compared to wheat or rice. You can see the strawberry production increase very rapidly in recently. It maybe suggests people want to start with strawberry when people will satisfy their basic food. And Japan, we also have many different types of strawberry varieties. And now more than 30 strawberry breeding stations in Japan, and the competition of variety is very high. And even so, strawberry breeding, their doing is quite simple because strawberry is propagated by runner, so you can propagate clones. Therefore, if the people find very good F1 individuals, they can propagate as these plants as a good variety candidate. So typical breeding strategy, they are just cross two parents and select good plants. And in Japan, strawberry breeding was started in the late 19th century, introduced the strawberries from the Europe, and then we keep improve the strawberry varieties. However, the used parent clone is a very not so large varieties. The number of used parent clone is limited. Therefore, the genetic diversity in the current strawberry variety becoming less. And in these situations, because of these situations, selecting good varieties by using conventional breeding strategy is becoming very difficult. And nowadays, many breeders are suffering to create more good varieties by using the typical breeding method. So now people considering maybe we are trying different mountains. So current varieties is maybe a top gene combinations. However, we use just limited number of gene combinations. So now it is a time to find out, to look for the new combination, gene combinations to create more great strawberry varieties. However, because climbing this mountain is very hard, so it is very tough work without no guide. Therefore, people are considering genetic selection in strawberry, and we have started genetic selection project in strawberry since 2015, and at the moment, 5 different strawberry breeding stations across Japan is participate in this project. And we use genome shuffling populations at the initial populations, and then improve these populations by using genomic selections. And this is a brief strategy of our genomic selection in breeding. So first, we make the population by crossing several parental clones and getting seedlings. And at the initial stages, we don't have any models. So the plant is transplanted to the field, and breeder doing trait evaluations. And at the same time, we are doing a whole genome-wide genotyping by using RAS-seq, and by comparing the traits and genotype data, we make a model, and then select the parents again. And next generation, now we get the model, so we are doing seedling selections. So after getting seedlings, we are doing sampling and do the genotyping for a large number of seedlings. Then a selected seed is transplanted to the field, and again the cycle is doing. And this is a more detail about each steps. So step 1 is a trait evaluation in the field and construct a prediction model. So as I told, we evaluate the traits in the field and doing the genotypings. And there's several different ways to comparison the genotype and the phenotype. One is a construction of the prediction model, and second is identification of SNP associated with the trait by GWAS. And also we use the another method to select a SNP for selection of individuals with target trait by names X. And this modeling is performed in each year because genetic structure of the each generation is changed by selecting the parents. And this is one the result of the model prediction, and we evaluate multiple traits and create each models for each trait, and use the these models especially for the models showing higher prediction accuracies. And this is an example of a GWAS, and as I told, we performed the modeling and GWAS analysis for each generations. It is because, for example, this is a food hardness GWAS result, and you can see the relatively higher peaks here in 2008. The traits are evaluated in the season of 2018 and 2019. However, this peak is disappeared in the next year. It is because this allele is favorable allele is selected in the next generations, and all generations have a favorable allele on this row side. That's why in the next generation, we cannot see the significant peak here. So because of this, we are doing modeling in each generations. And this is the second step. And once we create the modeling, we select the parents and make a crossing. And generally, each breeding station selects approximately 10 to 15 plants per populations. And one of the difficult points of this selection is we should select the parents which have very good values for multiple traits. But this is ideal, but in many cases, we cannot get such parent plants. So some plants have a good values in fruit size or fruit hardness, but some plants don't. But these plants don't have good traits in other traits, good values in other traits. That's why we should select the parents with the imaginations how we can get more good combinations in the next generations. So considering the trait balance is very important. And step 3 is seedling selection and transplant to the field. And in these cases, we should decide the markers you are using the seedling selections. And we generally choose markers which show a significant association identified by zeros or eggs. And again, at the time, we should put the balance of the target traits. And in our project, we use the number of used markers per population is approximately 30. So ideally, the number used in the seedling selection, if you use as many as possible in the ultimate ideally whole genome sequencing is best. However, at the same time, we should consider the cost and also periods. And because of a limited time, we should do genotypings within 4 weeks. Therefore, need high throughput and low cost system for these approaches. And at the beginning of this project, in 2015, we used SSR. This is because more SSR accepts crude DNA, which can extract low cost. However, SNPs require pure DNS. So we use SSR, but it takes a lot of times. Therefore, we introduce Octopure for this and getting a pure DNA with a lower cost. And currently, we extract DNS approximately 40 plant species and not only strawberries, but it works very well. And for genotypings, we use 2 approaches. One is CASP, and the other is a target umbilical sequencing task. And the accuracy is more, CASP is higher because sequences approaches are using bioinformatics, and their results sometimes changes by use bioinformatics tools. And when we use a typical real-time PCR for identify genotyping, high throughput and cost is almost similar. And even though we had very difficult situation at the beginning, because in our lab, approximately 300,000 reactions should be done in the during 4 weeks. And this is not only our work, So, and actually it is doing in the vacation season, but we have no vacation, and it's overwork, and the atmosphere is becoming very bad. So, so that's why we introduced IntelliCube for getting a relaxed times in our summer vacation and getting the summer vacation, and now we are doing a very good job with these machines. So this is a progress of our GIS project, and this graph shows the comparison of the 3 generations, and you can see the food hardness or food weight, and also bricks improved each generation. And from this generation, now we select the candidate plants for varieties. So this is a sort of genomic selection based on our experience. And the genomic selection is a method of selecting promising individuals based on large-scale data. And the use of appropriate data can dramatically increase the accuracy of selection compared to conventional breeding methods. And although it was initially difficult to realize, the introduction of automated analysis robots such as Octopia and IntelliCube has made it possible. And through the large geotyping data show useful information to breeders. And the final decision is made by breeders. So by utilizing new technologies, the possibilities for breeders can be further expanded. So this is acknowledgement. And this work is done by my team, and also 5 different breeding stations with many breeders, and we also have a collaboration with a similar university for genotyping and modeling. Thank you for your attention.
Thank you very much, Dr. Isobe, for the great talk on accelerating the strawberry breeding using the genomic selection technologies. I very much look forward to seeing those new varieties from Japan appear in some of our local supermarkets like Don Don Donki. I love those strawberries. So we do have a couple of questions. I was just looking through the Q&A in the chat. Yeah, so one question, just curious to get your thoughts about how do you think genomic selection can be routinely used, and what are the challenges that need to be overcome in order for genomic selection to be routinely used by the breeders? Can you share your expertise thoughts on this? What do you think are the most critical things that need to be overcome before it can be adopted routinely in the breeding program?
Yeah, I think the most important point is have a collaboration with a core laboratory, which can do in large-scale genotyping and modeling. So in many cases, breeders, they are good at field work, but not good at lab works and also bioinformatics. So I think the collaboration with each specialist is important for routine work in genomic selections.
Understood. So should be more collaboration of the breeders with experts like yourself and the Kazusa DNA research, that we can be able to have those nice varieties in our supermarkets coming soon. The next question, do you have any projects in planning? I know strawberry is interesting. Japan, has been very famous for all those fruits, nice fruits. Do you have any other plans to extend it into other species in your laboratory?
Yes, we also have genomic selection, especially for tree species. Tree means including fruit trees such as grape, apple, and citrus, and tea. And also we have non-tree species like buckwheat and sugarcane, etc.
Understood. Very much looking forward to that. The last question I have is with regards to imputation. Just wanted to get your perspective on the use of imputation, right? For some of those species that might have complex genomes, some of the recent trend is people are using imputation, instead of doing genomic selection, using tens, hundreds of thousands of markers, that with imputation, you're able to bring that number down to the lower thousands or even in a comfortable range, maybe of a 500 or a few hundred. So just wanted to hear your perspective or your experience using imputation, so that we can do a routine genomic selection, make sure it's affordable, it's cost effective in the higher upper hundreds to lower thousands.
Yes, especially for creating prediction models, imputation is very important. And so we, in our project, we established the bioinformatic pipeline. And in this pipeline, we do imputations, just input the VCF, initial VCF, and then this pipeline automatically do filtering and also imputation, and then created prediction models. So we use an in-house program, but also I think people can use the freely available imputation tools.
Great. Thanks for sharing your experience and expertise, Dr. Isobe. Very much appreciated. Arigatou gozaimasu.